Hey Team Bio, welcome to your first semester 2 screencast. For those of you who have watched these before, you know the drill. For those of you who this is the first screencast you are watching, this is very much like a lecture in class. I've got some visuals up here. You have a piece of paper with all the same visuals, and you're going to follow along as I write and talk. You will write down notes on your sheet, and you will have the sheet. You can bring it to class and you'll turn it in on Schoology, and there'll be an open note quiz first thing in class next period when this is due, and you'll be able to reference your notes to answer the questions on the quiz. And usually the quiz is pretty answerable if you've taken some good notes on this screencast. Yes, and this year this semester, we're gonna do something a little bit different. At the bottom of the screencast, you're gonna write me down one thing you're curious about, one thing you're still confused about, and doodle something that relates to what you learned in the screencast, but we'll talk about that at the end. Okay, generally I shoot for these screencasts to be between 10 and 15 minutes, but I'm talking a lot right now, so this one might be a little bit longer. Okay, so we are going to put together what we learned last semester with the Big Four and use it to understand the different components of the cell, which is the most basic unit of life. So we're moving on from kind of biochemistry and molecular biology into cellular biology and how cells, what they're made up of and how they function, which is made up of the things we just studied, and how they function is based in biochemistry. So it's all connected. Okay, so the cell membrane is made up of the Big Four organic compounds. And remember, the Big Four are fats and lipids, proteins, carbs, and nucleic acid, and UCL. Okay, and I'm gonna move this, actually, sorry, move it over here. The Big Four organic compounds. Okay, and the cell membrane is mostly made up of a certain type of fat that you have talked about, I believe, already. In my class you have, and I'm pretty sure in Mr. Falcone's class you have as well. They're called phospholipids. And this is their structure. They kind of look like a lollipop with two sticks. Okay. They're two fatty acid tails. Remember, these are long chains of carbon and hydrogen, so they are non-polar. And as such, they are hydrophobic. And you see here, there's one straight one. This is our saturated fat. And here we have an unsaturated, and this is, I believe, mono unsaturated. Yeah, saturated. Because you see it has one kink in it, which represents a double bond between two carbon atoms. And this helps, we'll talk about later, about with membrane fluidity. So remember, we talked about saturated fats are solid at room temperature, and unsaturated fats are liquid at room temperature. And this combination of saturated and unsaturated fats in the phospholipids helps give some fluidity to the cell membrane. Okay, and then remember, we talked about how all fats and lipids are hydrophobic. And we break the rules here with phospholipids. Phospholipids have a phosphate head. And this phosphate head is actually quite polar, which makes it hydrophilic. So this organic compound has a polar region and a non-polar region. So not all phospholipids are entirely hydrophobic, and the phospholipid is a perfect example of that. Okay, so how does this manifest? What happens when a phospholipid in conjunction with other phospholipids encounters water? And what happens is that they spontaneously form a structure called a bilayer. So if we put phospholipids in aqueous solution, which means a watery solution, they line up, they arrange themselves spontaneously in this way, where we have water here and water here, and the phospholipids arrange themselves so their heads are encountering the water on either side, and their tails are mingling together. And this structure is called a bilayer. And this is the lowest energy state for this group of organic compounds. So it will just happen if you mix phospholipids and water, it will happen spontaneously. So here we have two hydrophilic heads, and here we have hydrophobic tails. Okay, but you're probably wondering, well, what about these sides? Water is going to completely surround this structure, and then you're going to have water encountering these hydrophobic tails where the edges of this structure are. And you're right. So what actually happens is something like this, where now this is going to look remarkably like a cell, hopefully. So we are going to form a total, complete ring of a bilayer where we have water on the inside of this structure and water now on the outside of this structure. So this is H2O and this is H2O. And here you can see we have our bilayer. So the phosphate heads are all interacting with water on the outside of the structure and on the inside of the structure. And the hydrophobic interior of the bilayer is only interacting with other hydrophobic tails here. So this is my image that I created. It is helpful because it's like you just see this relationship between these are the phospholipids and here's the bilayer. But I think that this image actually gives you a sense of the three-dimensional structure that actually forms when you toss a bunch of phospholipids into water. So you create a cell. You've got water on the inside and water on the outside, and then your three-dimensional lipid bilayer. So here are individual phospholipids. We've got the heads, and then we've got the tails. Okay, so this, because of the structure of the cell membrane, the fact that it's got polar heads here and then non-polar interior, means that certain things can't get through the cell membrane. Essentially, things that are charged or polar have a hard time crossing the membrane. So we call the cell membrane is semi-permeable. Only certain things are passing through it. And we'll talk more in greater detail about what passes through the cell membrane and what is excluded from the cell membrane in a screencast, I think tomorrow or the next day. Okay, and the last thing I want to introduce to you guys is this idea that the cell membrane here, this image makes the cell membrane look like it is exclusively phospholipids, but that's not the truth. When you actually look at a real cell membrane of any cell, you'll see that it is what's called the fluid mosaic model. So this is a more realistic interpretation of what the cell membrane would actually look like. It's going to be studded with a bunch of proteins. So here we have some examples of protein channels. This is an H. So I said before, certain molecules, polar molecules, have a hard time getting through the cell membrane. So the cell can let them in by passing through a protein, a transmembrane protein, a protein that spans the membrane. Here we have some sugar, some carbohydrates. Studying this protein, and they're going to act as some sort of signal to other cells. So remember, we talked about the functions of carbohydrates. They can be short-term energy, they can be structural, and then they can also serve as kind of molecular flags. Well, this would be an example of a little flag that the cell is putting on. This carbohydrate structure is going to be telling other cells what this cell is. And then here is an example of a receptor protein. So a substrate is going to come in and bind to this protein, and that is going to cause a conformational change somewhere in here. And this transmembrane protein is going to kick off this protein, which is then going to go out into the cell. It causes a signal cascade. And then maybe there'll be some sort of change in gene expression. Okay, so we'll talk more about all of these ideas later, but I wanted to give you this idea that the cell membrane is not just phospholipids, it is a mosaic. And probably the only one of the big four that is not represented here is our nucleic acids. And even that, I'm not sure. We might argue that GTP, which we'll talk more about later, is involved. So maybe all the big four is represented in the cell membrane, even though I didn't talk about nucleic acids. Okay, and then this last idea, this fluidity part, the way that I've drawn these proteins is that they are fixed in this specific place. But you know that having this kink in this phospholipid is actually helping make it more fluid. It's going to be kind of, this phospholipid is not fixed into place. It can freely move around on this side of the membrane. So right now, this little phospholipid that I'm going to color yellow is here at T equals 1. But when I look maybe 5 minutes later, T equals 5, now the yellow phospholipid is over here. It's freely diffusing in the membrane. And the same is true for all of these proteins that are embedded within the membrane. They are not fixed in place, but instead they are moving around because all of these components are moving around. So not only is it a mosaic, which is studied with many different pieces, but it is also fluid. None of the components are fixed in place because of the fluidity of the lipids. All of the components are free to move around the cell membrane as well. Okay, so that is it for the cell membrane structure. Okay, what I want you to do now, this is new this year for folks who were in my class last term, is I want you to write down at the bottom of your page somewhere one thing you're still confused about. I want you to write down one question you're curious about. And I want you to draw me one doodle that relates to something you learned in the screencast. And then bring these to class with you, and we will talk about them and maybe show them off. Okay, so if you are doing this for the first time, you take a picture of this and you upload it to Schoology, and I'll see you all in class next week on Monday.